Hello everyone. My name is MaryLena, and I am a second-year PhD student from the Department of Statistical Science in collaboration with UT Southwestern's Medical Artificial Intelligence and Automation Lab. The work that I wish to present to you today consists of a novel piece of statistical methodology which we have devised as part of a larger project which uses AI to optimize radiation therapy for cancer treatment. You can read some of the technical details on the slide here if you're interested. The general form of radiobiology experiments goes like this. You inject a set of mice with cancer tumors, and then observe the tumor growth under the experimental condition of interest. However, in some cases, the mouse may need to be terminated due to extreme discomfort caused by the tumor. While this process is necessary for ethical reasons, it introduces some interesting statistical issues, in part due to the fact that mice with large tumors are more likely to be sacrificed. The currently used method for dealing with this phenomenon is called the last value carried forward technique, and it is exactly what it sounds like. You just take the tumor volume at the time of the animal's death and carry it forward to the end of the experiment. However, this method isn't great because it introduces a bias in estimation of the group means in the later portions of the experiment. So if you take the mean of these three curves at each point in time, there's going to be a false plateau here after the green mouse was terminated. The idea behind our method is that it is optimal to impute the missing data given two different pieces of information: the shape of the curve, which we can obtain from the other mice, and the starting sequence, which we observed before the mouse was sacrificed. Furthermore, it is desirable to obtain not only a point estimate, but some measure of variability of our imputed values, that is, how confident we are about our estimate, which allows us to create a range of plausible values for that point. The way that we do this is by obtaining not just a single value for each missing time point, but rather a distribution from which we can sample several plausible values. And once again, some of the details are available on the slide. We tested the performance of our method through computational simulation, where we know the true values of the underlying growth curve, visualized here by the thick black line. On this plot, the x-axis is time in days, and y-axis is tumor volume on the logarithmic scale, so LTV stands for log tumor volume. Here, each color represents a different simulated mouse, and the box plots illustrate the range of plausible values selected by our method. As you can see, they appear to capture the true trend, that is, the black line, fairly accurately. We also tested the method on a real dataset by deleting some truly known values and applying the method to see what it came up with. Here, the two plots represent two different mice as suggested by the mouse diagrams. In both cases, the mouse truly survived to the end of the experiment, but we pretend that it died at the third time point, applying the method and comparing to the true values in order to check the method's performance. The colored dots represent the true values, and the box plots characterize the values selected by our method. Once again, you can see that our method appears to capture the desired effect, a major improvement over the common practice. We have also made our method available as a software package for ease of access. Finally, here's an illustration of the method's usage in a real example. The two plots represent two different treatment groups of mice, a set of controls on the left, and a set who received immunotherapy only on the right. Once again, each color represents an individual mouse, and the box plots characterize the range of values selected by our method. Thanks for listening to my presentation.